Hello everyone, I'm Kenichi Kawano from Hiroshima University, Japan. Today, I would like to talk about this title, the curvature-sensing peptides function as a membrane interfactant that glues the small extracellular vesicles to the cell membranes and enhances vesicle cellular uptake. First of all, I would like to explain about small extracellular vesicles sEVs. They have the diameter from the 30 to 200 nanometer. sEVs are the biogenetic lipid nanoparticles secreted by most mammalian cells. They play an important role for the intracellular communications because they contain signaling molecules, DNA, RNA, and proteins and lipids. sEVs derived from the mesenchymal stem cells, MSC. They have the multiple functions, especially anti-cancer activity against the cancer cells such as HeLa and PANC1 cell lines. The idea in this study is that if we can develop the drug loading system on the sEVs, drug and sEVs could exhibit synergistic effect of anti-cancer activities. The conventional drug loading system on or in the sEVs as shown here. The chemical reaction, antibody recognition, genetic engineering, hydrophobic chain insertion, and electroporation are widely used. However, several challenges are remaining in the existing systems. For example, chemical reactions target the membrane proteins, but their expression level on the sEVs are very limited, and some methods are selectively is not enough. In this study, we use the curvature-sensing peptide to improve both the drug loading and the cellular uptake efficiencies, and we also tried to achieve the convenient and highly versatile method. In these studies, we designed the Dimeric curvature-sensing peptides named dimer NFAV5. This peptide is a homodimer of NFAV5, which was conjugated via the disulfide bond at the C-terminus. The N-terminus was labeled with small fluorescent dye NBD instead of the drugs. These peptides adopt alpha helical structure when they bind to the vesicles, and they have the hydrophobic phases and hydrophilic phases. This peptide recognizes the lipid packing defect on the surface and insert their hydrophobic phase into curved membranes. This is a saturation binding curve of dimeric form and monomeric forms. The dimeric form as shown in green remarkably improved their binding affinity compared with the monomeric forms. In CD spectra, the monomeric form convert from the random coil to alpha helical structure when then binding the vesicles. On the other hand, the dimeric form did not change their structure apparently, but probably they form a leucine zipper-like form in the solution and change to open form as shown here, suitable for vesicle binding. sEV isolation and DID labeling method. 2 years ago, we reported EV catch and release isolation system named EV-CaRiS and isolated EVs using this system. This method used the curvature-sensing peptide to capture EVs at acidic pH and wash out the larger EVs and other molecules. And after that, we changed the basic pH and release EVs from the peptides. To obtain the sEVs using fluorescent dyes, DID labeling was performed according to the previously reported by controlling salt concentration and performing the filtration. The particle distribution as shown here, after DID labeling and the peptide binding, slight change in the diameter was observed by the modification. Biomarkers of sEVs were detected by the Western blotting such as ALIX, heat shock protein 70 and SHG101 and CD63, and also DID fluorescent band was observed as shown here. The shapes of the sEVs was confirmed by 10 images. The ELISA data targeting CD81 revealed that the DID labeling and peptide binding did not affect the antibody recognition. We observed colocalization of sEVs and dimeric peptides by confocal microscopy. sEV and peptides were stained with DID and NBD respectively. They were mixed together and incubated for 10 minutes at room temperature in the medium, and then added into cells and incubated 1 hour at 37 degrees in the incubator. After that, we observed the cellular uptake using confocal microscopies. For Hubeck normal cells, no remarkable uptake was observed. On the other hand, Pank1 cancer cells, remarkable accumulation and cellular uptake was observed. The peptides are shown in green and sEVs are shown in the red. They accumulate at the same time on the cell surface, and the remarkable cellular uptake was observed as shown here. This analysis data show that the high colocalization rate between the peptides and sEVs. In this illustration, maybe the peptides recognize sEVs and they are binding to their cell surface at the football net. Enhanced cellular uptake of sEVs by peptides was observed for Pank1 and HeLa cell lines. At a low particle concentration of sEVs, 10 to the power of 7, sEV alone, no cellular uptake was observed. Similarly, the peptides alone were not taken up by cells. However, if the peptides and sEVs existed together, the remarkable cellular uptake was observed as shown here. So, similar phenomenon was observed both the Pank1 and HeLa cell lines. Cellular uptake was facilitated by the 5 to 6-fold higher for sEVs, and similarly, the 20 or 25-fold higher for the peptides when compared with each administered alone. To elucidate the mechanism of cellular uptake of sEVs and peptides, we hypothesized that electrostatic interaction between the modified sEVs and the negatively charged molecules on the cell surface, such as phosphatidylserine and heparan sulfate chains, are involved this step. Heparin was used as an inhibitor to prevent electrostatic interaction between the sEVs and these molecules on the cell surface. As expected, compared with the normal conditions, in the presence of heparin at the different concentrations, the peptides and sEVs cellular uptake was remarkably inhibited under these conditions. The reduction level of sEVs and the peptides was significantly decreased, and which was similar to the background level. To elucidate the mechanism why this complex is highly internalized, we hypothesized that modified sEV after binding to the cell membrane induce membrane curvature and recruit clathrin from cytosol. Clathrin is also a membrane curvature sensing protein involved internalization of sEVs. Under isotonic condition, a lot of fluorescent signals was observed inside cells. On the other hand, under the hypotonic conditions, the fluorescent signals were observed only the cell membrane but not inside cells. This meaning that under hypotonic conditions, water molecule enter to inside cells and the cell volume significantly increase as shown in the previous report. As a result, the membrane tension increased to prevent internalization of sEV to the inside cells. From this data, we can say that under isotonic condition, the modified sEVs induced membrane curvature for their internalization by recruiting clathrin proteins from cytosol. To determine the endocytosis pathway of modified sEVs, we used endocytosis inhibitors. This one is negative control and positive control, no treatment, and normal conditions. Blue one is macropinocytosis and phagocytosis inhibitors, APA, orthomanin, cytokeratin D. In the presence of these inhibitors, the fluorescent signals of the peptide and sEVs still observed, which was similar level to the normal conditions. On the other hand, in the presence of the clathrin inhibitors as shown in the red, pit stop 2 and the dinosaur, the fluorescent signals inside the cells was completely disappeared, as shown here. We tried again and again in the same experiment and obtained the same result, indicated that the modified sEVs and the peptides internalized together via this clathrin-mediated pathway. A similar result was obtained for the PANC1 cells. In the PANC1 cells, also clathrin inhibitors, as shown in red, the perfectly prevent internalization of sEVs and the peptides, as shown here. We revealed that the modified sEVs are internalized via clathrin-mediated pathway in both PANC1 and HeLa cells. This is a proposed mechanism of sEV cellular uptake enhanced by the dimeric peptides. sEV alone or the peptide alone, their cellular uptake is very limited. On the other hand, if they exist together as the first step, they form the complex and accumulated on the cell surface by electrostatic interactions. At the second step, the membrane curvature is induced by modified sEVs, and at the third step, they were internalized via clathrin-mediated endocytosis pathway. Here, the long side chain of lysine of peptides binding to the sEV surface could contribute to the electrostatic interaction with negatively charged molecules on the cell surface as shown here. We applied them for drug delivery system. Here, we prepared biogeneated dimeric peptide number 4 as shown in orange and biogeneated anti-cancer staple peptides as shown blue number 9 and number 10. The staple peptides are membrane impermeable peptides and they form the complex on the sEV surface via the structure adhesive as shown in yellow. We just mixed them together and incubated for 10 minutes in the medium and then added to cells and incubated 1 hour at 37 degrees. After that, removed this reagent and incubated again at 37 degrees for 23 hours. Number 9 and number 10, these staple peptides have the ability to induce the apoptosis of the cancer cells. Number 9 is SHP53-4 targeted HDM2 and released p53 proteins and resulting apoptosis induction. On the other hand, BMBH3, this activated BAX proteins, and these BAX proteins interacted with the mitochondria and release a cytochrome C release, resulting in the apoptosis induction. The left panel is anti-cancer activity and the right panel is caspase 8 activities. Black and white bar indicate the result of HeLa and PANC1 cell line respectively. Please focus on these red arrows. Under these conditions, peptide accumulated and formed a complex on the sEV surface. The cell viability of HeLa and PANC1 cell lines remarkably reduced under these conditions. Under same conditions, apoptosis indicator caspase 8 activity was also increased, demonstrating that this system was successful to induce apoptosis against the cancer cell lines. In contrast, number 9 and number 10, the staple peptide alone did not show strong anti-cancer activities because these peptides are membrane impermeable. We also confirmed that no membrane perturbation occurred for our system. This reagent is membrane impermeable. If the plasma membrane got damaged by this drug loading system, the nuclei will be stained by the propidium iodide. The methanol was used as a positive control. Propidium iodide stained the nucleus because the cell membrane was compromised or damaged by the methanol. On the other hand, these peptides did not induce membrane perturbation because no fluorescent signals of the PI was observed under these conditions. This meaning that treated cells with this system have intact membranes. In conclusion, the dimeric peptide exerted its function especially at low particle concentrations by highly enhancing endocytic uptake of sEVs via clathrin-mediated pathway. Cell uptake of sEV and peptide under coexisting conditions was facilitated in cancer cells by over 5-fold and 20-fold respectively compared with those administered alone. This system allowed us to load anti-cancer drugs onto the surface of sEVs and induce apoptosis in cancer cells. Finally, I would like to thank the member in the previous laboratory at Kyoto University. Also, I would like to thank the current member in the Professor Nagase laboratory at Hiroshima University. Thank you so much for your kind listening.